A growth medium or culture medium is a liquid or gel designed to support the growth of microorganisms or cells, or small plants like the moss Physcomitrella patens. There are different types of media for growing different types of cells. There are two major types of growth media, those used for cell culture, which use specific cell types derived from plants or animals, and microbiological culture, which are used for growing microorganisms, such as bacteria or yeast. The most common growth media for microorganisms are nutrient broths and agar plates. Specialized media are sometimes required for microorganism and cell culture growth. Some organisms, termed fastidious organisms, require specialized environments due to complex nutritional requirements. Viruses, for example, are obligate intracellular parasites and require a growth medium containing living cells. Types of growth media. The most common growth media for microorganisms are nutrient broths or LB medium. Liquid media are often mixed with agar and poured via sterile media dispenser into petri dishes to solidify. These agar plates provide a solid medium on which microbes may be cultured. They remain solid, as very few bacteria are able to decompose agar. Bacteria grown in liquid cultures often form colloidal suspensions. The difference between growth media used for cell culture and those used for microbiological culture is that cells derived from whole organisms and grown in culture often cannot grow without the addition of, for instance, hormones or growth factors which usually occur in vivo. In the case of animal cells, this difficulty is often addressed by the addition of blood serum or a synthetic serum replacement to the medium. In the case of microorganisms, there are no such limitations, as they are often unicellular organisms. One other major difference is that animal cells in culture are often grown on a flat surface to which they attach, and their medium is provided in a liquid form, which covers the cells. In contrast, bacteria such as Escherichia coli may be grown on solid media or in liquid media. An important distinction between growth media types is that of defined versus undefined media. A defined medium will have known quantities of all ingredients. For microorganisms, they consist of providing trace elements and vitamins required by the microbe and especially a defined carbon source and nitrogen source. Glucose or glycerol are often used as carbon sources, and ammonium salts or nitrate as inorganic nitrogen sources. An undefined medium has some complex ingredients, such as yeast extract or casein hydrolysate, which consist of a mixture of many chemical species in unknown proportions. Undefined media are sometimes chosen based on price and sometimes by necessity. Some microorganisms have never been cultured on defined media. A good example of a growth medium is the wort used to make beer. The wort contains all the nutrients required for yeast growth, and under anaerobic conditions, alcohol is produced. When the fermentation process is complete, the combination of medium and dormant microbes, now beer, is ready for consumption. Nutrient media contain all the elements that most bacteria need for growth and are non-selective, so they are used for the general cultivation and maintenance of bacteria kept in laboratory culture collections. An undefined medium is a medium that contains a carbon source such as glucose for bacterial growth, water, various salts needed for bacterial growth, a source of amino acids and nitrogen. This is an undefined medium because the amino acid source contains a variety of compounds with the exact composition being unknown. A defined medium is a medium in which all the chemicals used are known, no yeast, animal or plant tissue is present. A differential medium is a medium that includes some sort of added indicator that allows for the differentiation of particular chemical reactions occurring during growth. Example nutrient agar medium composition, beef extract, 0.3 Ag, tone, 0.5 Ag, NaCl, 0.5 Ag, agar, 1.5 Ag, distilled water, 100 ml, pH, 7. Minimal media are those that contain the minimum nutrients possible for colony growth, generally without the presence of amino acids, and are often used by microbiologists and geneticists to grow wild-type microorganisms. Minimal media can also be used to select for or against recombinants or X-conjugates. Minimal medium typically contains a carbon source for bacterial growth, which may be a sugar such as glucose, or a less energy-rich source like succinate, various salts, which may vary among bacteria species and growing conditions. These generally provide essential elements such as magnesium, nitrogen, phosphorus, and sulfur to allow the bacteria to synthesize protein and nucleic acid. Water. Supplementary minimal media are a type of minimal media that also contains a single selected agent, usually an amino acid or a sugar. This supplementation allows for the culturing of specific lines of autotrophic recombinants. Selective media. Selective media are used for the growth of only selected microorganisms. For example, if a microorganism is resistant to a certain antibiotic, such as ampicillin or tetracycline, then that antibiotic can be added to the medium in order to prevent other cells, which do not possess the resistance, from growing. Media lacking an amino acid such as proline in conjunction with E. coli unable to synthesize it were commonly used by geneticists before the emergence of genomics to map bacterial chromosomes. Selective growth media are also used in cell culture to ensure the survival or proliferation of cells with certain properties, such as antibiotic resistance or the ability to synthesize a certain metabolite. Normally, the presence of a specific gene or an allele of a gene confers upon the cell the ability to grow in the selective medium. In such cases, the gene is termed a marker. Selective growth media for eukaryotic cells commonly contain neomycin to select cells that have been successfully transfected with a plasmid carrying the neomycin resistance gene as a marker. Ganciclovir is an exception to the rule as it is used to specifically kill cells that carry its respective marker, the herpes simplex virus thymidine kinase. Some examples of selective media include, eosin methylene blue contains dyes that are toxic for gram-positive bacteria and bile salt which is toxic for gram-negative bacteria other than coliforms. YM is the selective and differential medium for coliforms, YM which has a low pH, deterring bacterial growth. McCunkie agar for gram-negative bacteria, hecton enteric agar which is selective for gram-negative bacteria, mannitol salt agar which is selective for gram-positive bacteria and differential for mannitol. Terrific broth is used with glycerol in cultivating recombinant strains of Escherichia coli, xylose lysine desoxycholate, which is selective for gram-negative bacteria, buffered charcoal yeast extract agar, which is selective for certain gram-negative bacteria, especially Legionella pneumophila, Beta Europarka agar for gram-positive Staphylococcus, differential media or indicator media distinguish one microorganism type from another growing on the same media. This type of media uses the biochemical characteristics of a microorganism growing in the presence of specific nutrients or indicators added to the medium to visibly indicate the defining characteristics of a microorganism. This type of media is used for the detection of microorganisms and by molecular biologists to detect recombinant strains of bacteria. Examples of differential media include, blood agar, which contains bovine heart blood that becomes transparent in the presence of hemolytic streptococcus, eosin methylene blue, which is differential for lactose fermentation, McCunkie, which is differential for lactose fermentation, mannitol salt agar, which is differential for mannitol fermentation, X-gal plates, which are differential for lac-operon mutants. Transport media should fulfill the following criteria: temporary storage of specimens being transported to the laboratory for cultivation. Maintain the viability of all organisms in the specimen without altering their concentration, contain only buffers and salt. Lack of carbon, nitrogen, and organic growth factors so as to prevent microbial multiplication. Transport media used in the isolation of anaerobes must be free of molecular oxygen. Examples of transport media include, thioglycolate broth for strict anaerobes. Stewart transport medium, a non-nutrient soft agar gel containing a reducing agent to prevent oxidation, and charcoal to neutralize, certain bacterial inhibitors for gonococcus, and buffered glycerol saline for enteric bacilli. Venkatraman Ramakrishnan, VR, medium for V. cholerae. Enriched media contain the nutrients required to support the growth of a wide variety of organisms, including some of the more fastidious ones. They are commonly used to harvest as many different types of microbes as are present in the specimen. Blood agar is an enriched medium in which nutritionally rich whole blood supplements the basic nutrients. Chocolate agar is enriched with heat-treated blood, which turns brown and gives the medium the color for which it is named. See also, cell biology, cell growth medium, references. External links, The Nutrient Requirements of Cells, Strategies for Protein Purification Handbook.